Assalamu alaikum, dear students. This is me, Atiq Ahmad. Today we are going to study the topic meiosis 2. Before proceeding towards the meiosis 2, we will have an overview of the previous lectures. We have started the meiosis process, the meiotic division. The meiosis basically consists of two major parts, meiosis 1 and meiosis 2. We have already studied the meiosis 1. At the start of meiosis 1, the interphase, which is similar both in mitosis and in meiosis, the interphase consists of G1 phase, S phase, and the G2 phase. In the interphase, during the synthesis phase, the number of chromosomes that remains 46, but the DNA is double to that of 92 chromosomes. So during the interphase of meiosis, when the DNA replication process occurs, so the quantity of DNA is doubled to that of 92 chromosomes, but the number of chromosomes remains the same, and that is 46. And the meiosis starts with the cell which contains the normal 46 chromosomes. So in the interphase, the quantity of DNA is double to that of 92 chromosomes, but the number of chromosomes remains the same, 46. And those 46 chromosomes have two chromatids each. So the number of chromatids are 92, and each chromatid have a DNA of one chromosome. So whenever the meiosis 1 occurs, so one cell is divided into two cells, and each cell receive the 23 chromosomes. Those 23 chromosomes have total chromatids of 46, and each chromatid have a DNA of one chromosome. So one cell is divided into two cells in meiosis 1, and each cell contains 23 chromosomes. Those 23 chromosomes have 46 chromatids, and 46 chromatids means 46 chromosome here, because the DNA in each chromatid is equal to one chromosome. Now we will move towards the meiosis 2. Diagrammatically, we will have an overview of the meiosis 1, and then we will proceed toward the meiosis 2. Now here, this is the cell, the prophase stage of meiosis 1. Before this prophase stage, already the interphase has already occurred. The quantity of DNA is double to that of 92. The chromosome number will remain the same, 46, but the number of chromatids for 46 chromosomes, those are 92, and each chromatid have the DNA equal to one chromosome. So whenever this cell is divided into these two cells, so these two cells have 23 chromosomes each, but the 23 chromosomes have 46 chromatids. This cell have 23 chromosome, and those 23 chromosomes have 46 chromatids, because each chromosome have two chromatids, and those 46 chromatids each have DNA of one chromosome. Now these two cells will enter into the meiosis 2. So this one cell will enter through this way, and this will move through this way, and this cell will be divided into these two cells, and this cell will be divided into these two cells. So at the end of meiosis 2, 4 cells, to these and to these 4 cells will be produced, and each cell will have half number of chromosomes, half number of chromatids basically. So that is known as haploid cell. So we will move into the details of the meiosis 2 now. Introduction. Meiosis 2 is the second part of meiosis and is similar to mitosis. This is similar to mitosis. It consists of prophase 2, metaphase 2, anaphase 2 and telophase 2. Now the teaching points. Prophase 2. The chromatin condenses and chromosomes become visible. At the end of meiosis 1, the chromosomes are again uncoiled and they are converted into chromatin network. But whenever the meiosis 2 starts and the prophase 2 starts, the chromatin again condenses and chromosomes become visible. Whenever you observe the chromosomes under the microscope, so they are properly visible. Each chromosome consists of a centromere and 2 chromatids. Now remember, each of these two cells, they have 23 chromosomes, this one and this one, and each chromosome have 2 chromatids, so 46 chromatids are there, and each chromatid have DNA of 1 chromosome. So each chromosome consists of a centromere and the 2 chromatids. During the prophase, the nucleoli and nuclear envelope will disappear. This is as usual. We have studied in mitosis 2 that in prophase the nucleoli and nuclear envelope disappear, and same was the case there in the prophase 1 of meiosis 1. Then centriole will move to the opposite pole and make spindle fibers. Again, this is similar to that of prophase 1 of meiosis and similar to the prophase of mitosis. In the metaphase, again this is similar. The chromosome attach with the spindle fibers, or in other words, the spindle fibers attach with the chromosomes at the centromere position and arrange themselves along the equator of the cell, forming an equatorial plate or metaphase plate. This is similar to that of mitosis and meiosis 1. Now diagrammatically we will have a look of the diagram here. This is meiosis 2. So here the chromosomes are arranged at the equator. These are the chromosomes. This is the metaphase stage, and these chromosomes are arranged here at the central region of the cell, which is known as equatorial plane or equator, and they form the metaphase plate. This is known as metaphase plate, where the chromosomes are arranged at the center of the cell, and same is the case here. Now remember, two cells are dividing now through the process of meiosis 2, and these 2 cells are already produced in the meiosis 1. Now these are centrioles. They move towards the opposite poles. This is one pole, this is another pole, and this is one centriole, this is another centriole, and these spindle fibers will attach to the centromere of these chromosomes. So here it will be attached, from this side here it will be attached, and same is the case with the other chromosomes. So in the metaphase, the chromosome arrange themselves along the equatorial plate or equator and form equatorial plate or metaphase plate. Anaphase, again similar to mitosis and that anaphase 1 of meiosis, the centromere of each chromosome divides, splits due to the pull exerted by spindle fibers. Now the spindle fibers which are attached with the centromere of chromosomes, they will shorten and they will exert a pull force. So they will pull the chromosomes. As a result, the centromere will split and each chromatid will move towards one end and towards the other end. So in the anaphase, the centromere of each chromosomes divide, splits, and why it is splitted? Due to the pull exerted by the spindle fiber. Because the spindle fibers shorten, they contract, and they exert a pull on the centromere of chromosomes. So the centromere will split and the two chromatids will be separated, and half of the chromatids will move this way and half of the chromatids will move this way. So half of the chromatids will move towards the one pole and half of the chromatids will move towards the other pole. Now we will move towards the telophase. In the telophase, the sister chromatids of each chromosome reach to the respective poles or opposite poles. The half chromatids will move towards the one pole, the half will move towards the other pole. In the anaphase, they have started their movement, and in the telophase, they will reach towards their respective poles. Half of the chromatids will reach to one pole and half will move towards the other pole, half will reach to the other pole. The nucleoli and nuclear envelope, nuclear membrane will reappear. This is similar to mitosis and as that of meiosis 1, that the nucleoli and nuclear envelope, it will reappear. It is usually the reverse of prophase. So in the prophase, they were disappeared, now they will reappear. So the nucleoli and nuclear envelope will reappear. And at the end, what will happen? The cytoplasm then divide to form 4 haploid daughter cells from the 2 cells entering into the meiosis 2. The 2 cells which have entered into the meiosis 2, those will be divided into 2. So one cell will be divided into 2 and the other will be divided into 2. So as a result, 4 cells and 4 cells with haploid number of chromosomes that will be produced. Diagrammatically, it is shown here. This is the metaphase where the chromosomes arrange themselves on the equatorial plate or on the equator at the central region of the cell, forming this metaphase plate. Then this is anaphase. The chromosomes are pulled, the centromere will split, and half the chromatids move towards one pole and half will move towards the other pole. In the telophase stage, the chromatids will reach to their respective poles, and then the cytokinesis will occur and the infolding, this is known as infolding, this cleavage will occur in between the 2 cells, and as a result, this one cell will be divided into these 2 cells, and this one will be divided into these 2 cells. So as a result, 4 cells will be produced, and these 4 cells now have half the number of chromosomes, and that is 23. So this is all about today's lecture. See you next time. Take care. Allah Hafiz.